Hello guys, today I will be teaching you DNA replication. DNA replication is a part of genetics. Genetics is a topic which is introduced in biology in 11th 12th class. So let's begin. In at least in India, in other countries it will be different. DNA is deoxyribonucleic acid, right? And RNA is ribonucleic acid. DNA and RNA consist of the genetic material of a cell. So DNA deoxyribonucleic acid, RNA is ribonucleic acid, and they both are genetic material in a cell present in the nucleus of a cell in eukaryotes. So this is specifically in eukaryotes, right? And in prokaryotes, the thing is that there is chromatin form present in the cell freely. So there is nucleoid. So present in nucleoid in the case of prokaryotes. Now let's talk about central dogma of molecular biology. So central dogma of molecular biology consists of DNA, RNA, and proteins. Right. So DNA can make copies of itself. DNA can make copies of itself, which is called as replication or duplication of DNA. So replication of DNA or duplication of DNA. And DNA can also be formed to RNA. RNA is single stranded and DNA is double stranded. There is slightly difference in RNA and DNA, but not much, right? So transcription is the process through which DNA is converted to RNA, and reverse transcription is the process through which RNA is actually converted to DNA. So DNA can also be converted into RNA, and RNA can also be converted into DNA. And RNA can be translated to proteins. So RNA can be converted to form proteins through the process of translation. But there is no reverse of translation. No reverse translation. Proteins cannot be converted back into RNA. So proteins cannot be converted back into ribonucleic acid, but RNA can be converted back into DNA. So this is the central dogma of molecular biology, which is kind of very important. So let's talk about replication of DNA. Replication of DNA consists of two types. First is prokaryotic cell and eukaryotic cell. Eukaryotic cell consists of the linear and double stranded DNA, and prokaryotic cell consists of a circular and double stranded DNA. Double stranded is also represented by DS sign. So DS can be represented for double stranded. So linear DS DNA and circular DS DNA. In prokaryotic cell circular, in eukaryotic cell linear. Linear means straight. So in the circular DNA of prokaryotes, there is a single origin of replication. So what is origin of replication? Origin of replication is a point from where the bubble forms and they start to separate. So both the strands start to separate from origin of replication. Or origin of replication is also represented by ORI or ORI. So ORI, if anyone tells you about ORI, you mean origin of replication. And it has a single origin of replication. So there is a single point from where the DNA strands start to separate. And in the case of eukaryotic cells, so in the linear DNA, there are multiple origin of replications. Why is that? For faster process, cause the DNA is a very, very long chain. So for becoming faster, they have to create multiple origin of replications. But the DNA of prokaryotic cell is not that big. So that's why there are multiple origin of replication or multiple ORIs. Okay, so let's talk about why and when do we need do we need a replication? Why replication? Right, replication / duplication. So why do we need it and when do we need it? So here is, I have made a diagram to tell you that. So there is 4 phases in a cell. One is a mitotic phase. You might be knowing mitotic phase. Mitotic means that one cell divides into two cells. And there are two growth phases, G1 phase and G2 phase. Both are growth phases where the cell grows and ribosomes are formed, many stuff happens. And there is an S phase. S phase is also called as replication phase. So there is an S phase, G1 phase, G2 phase, mitotic phase. And in mitotic phase, what happens is the chromosomes remain same in daughter cells. So there is a mother cell and two daughter cells are formed. So the chromosomes remain same in the daughter cell, but the DNA is halved. DNA is halved in each daughter cell because the DNA is fixed. So that's why in S phase replication phase, chromosomes still remain same, but the DNA is doubled. So once they were halved, so now it is doubled. So now they can perform as a mother cell. So by growth phase after G2 phase, it can again start mitotic phase. So that is why replication is needed. And when does it happen? Replication of DNA occurs in S phase or the interphase. G1 phase, G2 phase, and S phase consist of interphase, and mitotic phase is M phase, which where the cell division happens. So now let's talk about DNA replication, right? So DNA consists of two strands. This is the unwinded diagram of a DNA strand. It consists of a 3' end. 3' is basically defined as the free end of a carbon chain. So 3 end is the free end of a carbon chain in a sugar. So there are basically multiple sugars. So a sugar here has 3 end, 5' end, and it is connected to the 3' end of the second sugar. So it is connected to the 3' end of the second sugar, then 5' end of this, and so on. In the end, there is a 5' end. And the second strand is complementary to that. So the 5' end, so here there will be 3' end, then sugar, then 5' end. 5' means the location of the carbon, which through which everything is connected or free, right? So 5' end, one 3' end is connected to a 5' end, and vice versa. So let's talk about oris. There is an ori created here. There are two oris I have made. So oris form replication bubbles. These things which form by the action of ori. How does an ori create? So it is created through helicase enzyme. Helicase is an enzyme which works to say unzipping enzyme. So it unzips both the strands and basically separates it. How does it separate it? By breaking the hydrogen bond between the nitrogenous bases. So it breaks the hydrogen bond in the nitrogenous bases and it separates them. Right. So where it would start separating, there will be a 3' end of a sugar. Let's say there is a sugar here. So 3' end, and there is a 5' end on the opposite side. So 3' end will be connected to 5' end, then there will be a sugar, then 3' end here, 5' end here, like that, right? So there are two strands. One is leading strand and one is lagging strand. So leading strand is this, and leading strand is this, and the opposite side. So this is a leading strand. This will be a leading strand, right? And these opposite sides, so this and this, these both will be lagging strands. So this is a lagging strand, and directly opposite to it is the another lagging strand. So why is that leading and lagging? I will tell you later. And there are multiple oris happening. Replication bubbles are forming. Leading strand is said to be a continuous strand. So continuous formation of strand is happening, and lagging strand is said to be a discontinuous strand. So continuous strands are not being formed. So discontinuous, right? So this is shaping like a fork, right? Bubble is shaping like a fork. So this is called as a replication fork. So an helicase enzyme forms a replication fork, right? So, and let's talk about SSBP. SSBP full form means a single stranded binding protein, and it is evident from the name as well. So what it does is that it binds to this to both the strands, and it keeps them from joining together again. So if it is not present, they might join even together. The hydrogen bonds might join together again. So it keeps them from joining. Right. So that is the function of SSBP. Now there is an enzyme called as primase enzyme. Primase enzyme is, what does primase enzyme does? It forms a primer. Now what is a primer, you might ask? A primer is a small strand of nitrogenous bases nitrogenous bases of RNA. So RNA, if you know that RNA has different nitrogenous bases than the DNA. DNA has thymine, right? But RNA has uracil instead of thymine. That's the only difference, but there is a significant difference between RNA and DNA. So primer is an RNA strand. It is not a DNA strand which joins with the replication fork in this. So here let's talk about primers. Primer is something which joins only with the 5' end. Means basically you can say primer joined here. So primer will join with the 5' end here, and this will also have 5' end like sugar, and end it will be 5' end. So the work which is done of joining the bases, nitrogenous bases, is basically done by DNA polymerase 3. DNA polymerase 3. This is what enzyme is used to, building enzyme it is said. So it is used to join different nitrogenous bases, right? So this enzyme builds new nitrogenous bases, but it needs a primer to start. So DNA polymerase 3 needs a primer to start, right? So what happens in lagging and leading strands? Let's say this is a 3' end, so opposite it will be 5' end. By I said complementary strands, so opposite it will be 5' end. So a primer will be attached to these sides, right? A primer will be attached here. So here there will be some primer, and here there will be some primer, because this is also a 3' end. So here will be a primer, right? So there will be 2 primers in the leading strand, and then it will DNA polymerase 3 will start working, and it will start joining new nitrogenous bases. So this starts joining. So this is continuous. What is the main rule? There is a main rule that DNA polymerase 3 only works from 5' end to the 3' end. Now what am I saying, 5' and 3'? This primers 5' end to the 3' end of this template strand. These both strands are called as template strands. So basically these both will be said as template strands, right? Template strands. So primer, so 3' end of the template strand and 5' end of the primer. So basically what I'm trying to say is 5' end of the primer and 3' end of the template strand. This is the template strand. So the DNA polymerase 3 only works from 5' end to 3' end. So it will join these enzymes continuously. That's why this is given the name leading strand or continuous strand. But what will happen in the other case? The thing that will happen is that the primer cannot be formed here. The primer cannot be formed here. Why cannot not be formed here? Because the DNA can only go from 5' end to 3' end. But here if you take any point, it will have a 5' end, right? this sugar will, at any point will have a 5' end, and that at the template strand. So at the template strand there will be 5' end, but the DNA polymerase 3 cannot actually work from 5' end to 5' end. So 5' of primer to 5' of template cannot work. So what actually happens in this case is that the primer is actually formed in this part, not in basically this part, like not in the starting of the ori, but uh, the sides of the ori, and there are multiple primers forming. Not only one, but there are multiple primers forming, right? So there will be formation of multiple primers, and this will contain 5' end. And now if they go to this side, now if they continue to this side, right, then they will only reach to 3' end. I have written here, explained here. So 5' end here, so this will be 5' end here, and here will be 3'. Now this will be 3', right? So that is how lagging strands are created, and this is not created completely. So there are Okazaki fragments formed. What is an Okazaki fragment? An Okazaki fragment consists of Okazaki, I don't know, it's some sort of Chinese name. Okazaki fragments. So it consists of a primer plus the bases, the nitrogenous bases formed. So it consists of primer and the nitrogenous bases, which are actually formed by this primer plus the nitrogenous bases formed by that primer. So this will be have a 5' end, and this will have some 3' end. So this will have 3' end, and here 5' end. So this can work. Here this to this direction can work. This to like in from left to right it cannot work, because that will be 5' to 5'. But from right to left it can work, because that will consist of 5' to 3', right? So that will consist of 5' to 3', so that can work. So there are multiple primers added, and then they complete like that. This goes to there, then this goes to there, then there will be another primer here, then this goes to there. So that is how it is completed. But the problem here, still there is something problem, and the Okazaki fragments, like this Okazaki fragment, then this Okazaki fragment, there will be this Okazaki fragment, then there will be multiple Okazaki fragments, right? So they will be joined by DNA ligase. DNA ligase or molecular glue, which is said, joins the Okazaki fragments together, right? So basically they join together, and the primer is removed at the end. Why is primer removed? Because it consists RNA. If you remember, I wrote that RNA consists of a primer, but we cannot have RNA bases, like we cannot have uracil in the formed DNA, right? So they are removed, primers are removed, and DNA polymerase 1, there was DNA polymerase 3 which was making these strands, this continuous strand. Most of the work is done by DNA polymerase 3, but to join the bases done by primer, to join the space left through the primer, DNA polymerase 1 is required, which fills these remaining new strands, and thus you can create 2 strands, right? This complete strand done, and this complete strand done. So what you did was replication. If you see clearly, here there was only one strand, means there was only one DNA molecule, right? Now there are two DNA molecules, two daughter DNA molecules. So that is how DNA replication is done. Thank you. Bye bye.